Welcome back to Bio 255 Microbiology Lab Session 5. Please turn to Session 5, page 1 in your lab manual. Today we are going to perform simple stains. If you recall from the last session, we viewed living organisms under the microscope. And the problem with viewing living cells using compound light microscopes with bright field microscopy is that the background is almost as bright as the cell itself. So there's very little contrast between the living cell and the slide background. That makes it difficult to view. To improve the ability to view bacterial cells, we are actually going to stain them using microbiological dyes. Before we get started, let's take a look at your learning objectives for this session. First of all, I want you to be able to properly prepare bacterial smears on slides for staining purposes. In addition, I want you to follow simple staining protocols in order to increase the contrast between bacterial cells in the background so that we can view them more readily under our microscopes. You should be able to explain the chemistry behind simple stains and behind both of the types of simple stains which we are going to do today. One is called a simple basic stain and the other is a simple negative stain. So I do expect you to understand how these staining procedures work at the cellular and the molecular level. And finally, I want you to utilize the proper terminology for describing bacterial cellular morphologies and cellular arrangements. Let's take a look at the two simple stains on the docket for today. We are going to do these simple stains using a single dye. In fact, that is what the definition of simple stain is. A simple stain uses only one single dye. We have two types of simple stains that we will carry out. The first, as I mentioned, is a simple basic stain. And a simple basic stain uses a basic dye. A basic dye is a positively charged dye molecule. Today, we are going to use Crystal Violet from your dye box in your drawer. Crystal Violet is a purple dye. It is positively charged. We will also do another set of simple basic stains using safranin. Safranin is a pink dye which is positively charged. Both Crystal Violet and safranin are called basic dyes. They contain a positive charge. When we stain our bacterial cells with either one of these dyes, the positively charged dye molecule adheres to the negatively charged cell surface. The cell walls and cell membranes of bacterial cells are negatively charged, so they attract basic dyes. The second stain that we are going to use is called a simple negative stain, and it is going to use the dye known as nigrosin. Unlike safranin and Crystal Violet, nigrosin is a negatively charged dye molecule. It will not adhere to the cell surface. It will actually repel it. So that is the simple negative stain dye for the day. I'm going to move your dye box out of the way, and we are going to take a look at the bacteria that we are going to use in this lab session. So at this point, we should talk again about the genus and species of bacteria. When we name our bacteria, we always give the genus. We know it is capitalized, and the species, and we know that's all lowercase. I have here our 4 stock bacteria for the day, and we are going to use Staphylococcus aureus. The genus is Staphylococcus aureus. We are going, is Staphylococcus, and the species is aureus. We are going to use something called Escherichia coli. We are going to use Bacillus subtilis and Rhodospirillum rubrum. These 4 names give us clues to their shapes. At least some of them do. Now, Escherichia coli does not tell us a lot about the shape of the cell. But when we look at the name Staphylococcus aureus, Staphylo means grape-like, and coccus means spherical. So right off the bat, we are going to expect to see spherically shaped cells in grape-like clusters when we're done staining and we view our slides under the microscope. When we consider our stock known as Bacillus subtilis, the genus Bacillus tells us it is a rod-shaped bacterium, so we know what to look for. And finally, Rhodospirillum rubrum. That genus, Rhodospirillum, tells us it is a spirilla. Spirilla are spiral-shaped bacteria. Rubrum, by the way, means red, and you can see up close on this stock culture that this organism produces a red pigment. Okay, so those are our 4 stocks. Let's go ahead and get started. The first thing I'm going to teach you to do is to make bacterial smears. To do this, I'm going to lay out 4 slides for our simple basic stains. One set of 2 slides we are going to stain with crystal violet. We're going to do the identical smears on the other set and stain those with safranin. The first thing I want to do is properly label these slides. So I'm going to divide them in 2 because I'm actually going to place 2 bacteria on each slide. On the first slide, I'm going to put Staph aureus on one side and Escherichia coli on the other. Notice I'm using a 2 initial system here, SA meaning Staph aureus, EC meaning Escherichia coli. I said we are also using Bacillus subtilis. It has some unfortunate initials. And we are going to use Rhodospirillum rubrum RR. So I am going to label these on the other set as well very quickly. Notice I'm using a grease pencil because that wax stays attached to the slide no matter what. Okay, now I'm going to make some bacterial smears. When you make bacterial smears, we're going to come over to the sink just a second. I'm going to actually just grab a loopful of water in my loop. Tap water is fine. And I'm going to place that on my first slide. I'm going to put a dollop of water in the middle of each of the halves of that slide. I need to put my goggles on, light my fire, and now I am going to grab Staph aureus, my first stock. Flame my loop to sterilize it, go into my stock culture, cool it down, and grab some bacteria. Now, this is a tricky bit for students when they first learn to make smears, because if you grab too much, you are going to make such a dense smear that we will be unable to resolve individual cells. So the key to making a good bacterial smear is, first of all, don't grab big chunks of bacteria. And secondly, when you add it to the loop full of water, spread it out over the entire area of the slide, at least where there is no label. I'm going to let that air dry, but did you notice that I've spread that entire little loopful all around that half of the slide? Okay, I'm going to flame my loop. I'm going to go back into our next stock named E. coli. Escherichia coli, everybody's heard of, simply referred to as E. coli. Put that back in the rack, and I'm going to drop that down into the loop full of water. Again, I'm going to spread it all over the surface on this half of the slide. Notice that the water is a little bit turbid. Okay, so I know I've got bacteria in there. I'm going to go ahead and I'm going to put drops now on all of my slides. I'm going to make the smears for both the crystal violet set of simple basic stains and for saffronin set. So a loop full dropped on each half is all we need. The reason I use a loop and not a dropper is that one of the biggest mistakes students make is they put too much water on the slide to begin with. And then the problem they have is that it takes forever to dry, and you have to allow your smears to dry before you can heat fix them, and they have to air dry, and that can take quite a long time. So I'm spreading our cells out here. I'm going to put some E. coli on this. Again, making sure I don't make the smear opaque. You want to be able to see through your smears when you are done. That tells you that you're spreading the cells apart well enough that you can see them, but you also have a thick enough smear that you'll be able to find it under the microscope. Okay, let's move on to Bacillus subtilis here, and let's go ahead and grab some Bacillus subtilis and drop it on this slide. Spread it out. You can see that dropper full is already starting to dry. Bacillus subtilis is textured quite brittly. It's very brittle, and so you can see these chunks of cells. And I'm just going to grab some from one slide and move it over to the next. Spread that. And then rhodospirillum. Take my last stock culture and I'm going to go down into the auger, cool it down. You may be able to hear that sizzle. On this, I'm going to grab some of this red material here, bring it up, and I do have quite a bit of rhodospirillum on here. I want to spread that very, very thin. I'm going to, in fact, flame my loop and cool it down again so that I do not burn the cells, but I'm going to spread them out, again getting a nice thin layer. It looks as though I have a chunk of agar there, and that's something you want to avoid when you make bacterial smears. You don't want to bring agar onto the slide. It's hard enough to find the bacterial cells when you're looking at spicules of agar and trying to decide if that's what you should be focusing on. It can be a little bit frustrating. Okay, we're going to let those 4 slides air dry. And as they air dry, I'm going to put them aside along with our crystal violet and our saffronin. And while we're waiting for those to dry, I am going to do the negative stain. For this, I'm going to go ahead and get gloves on. Once I start working with microbiological dyes in this class, I'm going to always wear gloves. So I will put my gloves on, and I'm going to show you what we are going to do. The simple negative stain simulates dark field microscopy. I am going to put Rhodospirillum rubrum on one of these slides, and I'm going to put Staphylococcus aureus on the other one. Now, those are the only 2 stocks I'm going to use. I'm going to need 2 additional slides, as you'll see in a moment. I'm going to put about a ½ an inch in from the end. I'm going to put a drop of nigrosin on each of these slides. Into that drop of nigrosin, I'm going to add my stock cultures. So I'll grab Staph aureus first, Flame my loop, of course. Cool it down. And I'm going to grab a little bit of staff on the end and tap it into this Garrison. I am not spreading the Garrison. I want to keep it as a drop. And you'll see why in just a moment, but I'm going to tap off all those cells that I just picked up. I'm going to flame my loop again and get the Rhodospirillum rubrum. Okay, here's the Rhodospirillum rubrum. Cool my loop down. Grab some RR. And I'm going to tap that into the Garrison droplet. Now I have my bacteria in the Garrison. Flaming the loop. Putting that down on the bench. So what do I do now? This is why you need the second slide. When you use Garrison, you are trying to black out the background. To black out the background, you actually take the second slide and you spread it across the original slide like that. Now keep in mind, this right here is contaminated. The edge of that is contaminated. So what I'm going to do is I'm going to walk this slide over to our slide disposal. In fact, I'll pick this up and bring it back to the bench with me, but whenever you have contaminated slides or you're done viewing your stained slides, you simply dump them in this container. So we'll put this over here to keep it handy, and let's go ahead and let's make the smear on Staph aureus. Now on the first one I pulled it, but you are also welcome to push it like you would a blood slide. And that's it. We're done with the negative smear. You do have to let this air dry. So we'll move that out of the way. We will let those air dry. And I'm going to come back to my simple basic stains. They have now air dried. With my simple basic stains, I need to do something called heat fixing before I place the dye on them. I'm going to use the slide holder. You pinch it to open it up and you insert the slide between the two grips. And now, to heat fix it, I'm going to run it, 3 or 4 times very quickly through the meeker burner. Now notice, I didn't let it sit in the middle of that flame. If I did, I would caramelize those cells and that would be a mess. What I'm trying to do is I'm trying to melt the cells, so to speak, to the glass so that they stick. It also kills the cells, but every one of these cells has a little bit of a diffuse slime layer made of polysaccharides or glycoproteins on the outside of their cells. And when I heat that, like sugar, when it's melted, it sticks to the surface. So these cells are now stuck to the glass. Why did I need to do that? I needed to do that because if I didn't and I added these water-based dyes, the cells would simply wash right off the slide. So here I go again, heat fixed. We'll do this again quickly for our other set. And again, it is so rapid. 1, 2, 3, 4, I'm done. That's all you have to do. Final set here. 1, 2, 3, 4. I am ready now to stain. I do not need this flame on anymore, so I'm going to take that off. I'm going to take my goggles off once I've turned off the gas. Over here at the sink, I have my slide rack. I'm going to go ahead and I'm going to place each and every one of my slides on the rack for the simple basic stain. The simple negative stain is still air drying. Now, One set of these slides, I said I was going to stain with the basic dye known as crystal violet. Whenever you stain a slide, you stain the entire slide. You cover the entire thing with dye. You don't miss any part of it. You want to make sure the entire smear is covered. Okay? On this second set of slides, see, I got a little drop of crystal violet on that, so I'm going to wash that off. On the second set of slides, I'm going to add safranin, our second basic dye that we are going to work with. And you notice the color here is pink. Obviously, crystal violet tells you it's going to be purple. Safranin is pink. These are two dyes we are going to work with later in the semester to do differential staining rather than simple staining. And the first stain we're going to do is called a gram stain, and we'll use both of these dyes at that time. Okay, so we let those dyes sit. 60 seconds is sufficient. In fact, about 30 seconds is sufficient, but you can let it sit as long as you want. The length of time that the dye sits on there is not going to matter. As long as it's more than 30 seconds or so, you know you got the cells stained. So we could let it sit on here for 2 minutes. We could let it sit for 5 minutes, 10 minutes, not going to make a difference. What you don't ever want to have happen, though, is you never want your dyes to dry out. So 1 minute is good for these. I'm going to go ahead right now, and I've got my gloves on, so I can simply pick these slides up, and I'm going to rinse them under the water. And as I do so, let's take a look against my white lab coat and see, can you see the smears of bacteria that we made? Sure enough. So rinsing those, it's always good. I'm going to grab a paper towel here. It's always good when you have rinsed your slides to dry them off. To dry a slide off, you want to completely wipe the bottom of the slide, but you never wipe the top. So how do you dry the top? You dab it. You set it down on a paper towel, and you just tap it like that. And voila, that slide is ready to be viewed under the microscope. So let's go ahead and do that with these others. Good rinse. Don't ever forget which is the top of your slide, because that is going to be a problem if you put it upside down on the microscope. And here are the safranin slides. Let's take a look at those. I think you can see that the safranin stained those smears pink. Okay? Wipe that and tap it dry. And here's our final slide. Here we go. I'm going to use this paper towel to wipe the bottom off and tap it. We have a nice, large, obvious pink smear there. Okay, so let's go back to our simple negative smears. Here is Rhodospirillum rubrum. Here is Staphylococcus aureus. They are now air dried. The thing about a simple negative stain is you do not need to heat fix it. In fact, heat fixing will ruin it. So we are simply allowing these to air dry, and then they are ready to go on the microscope. At this point, if I were to run this under the water, it would wash the smear right off, because remember, these cells are not stuck to the slide. So what I've got here are 6 slides, and this set right here is a set of simple, basic stains using crystal violet. This is a set of simple, basic stains using safranin. And this is a set of simple, negative stains using nigrosin. These are heat fixed, these are not. Okay. So I'm going to meet you at the instructor microscope and we'll have a look at these slides. Let's just do a quick review of how we are going to focus all this. Remember, you're starting on your scanning power red lens and the stage is all the way up. And the first thing we have to do is we have to try and center in the middle of the white light coming through the stage something. We got to find something. And so we look at that slide and we have a purple smear. This is the purple smear that corresponds to the Staph aureus smear. And so I look at the slide, I find a big clump of purple. That's a good place to start. I put it in the middle of that center of white light coming through the stage. I'm on my red scanning power lens, and now I've got the stage all the way up. What do? I focus down with the coarse adjustment knob. And what do I see? A big blob of purple. This is not going to allow us to discern cells well, but it gets us a start so we know we're in the focal plane. At this point, I have already, and I suggest you do this as well, I've already placed a drop of oil on each and every one of my slides. Because remember, once we get one slide in focus, on oil immersion, we never lower the stage again. We simply leave it there and we slide the slides out and put the fresh slides on, and we know we're at the right focal point. So here we go. This is kind of in focus. I'm going to use a fine focus, make it a little cleaner, and then I am going to go from the red lens to the white lens. And it's not in focus completely, but parfocal tells me I ought to be able to just tweak the fine adjustment knob and it comes into focus. And what do we see? So right now in your lab manual, I want you to draw Staphylococcus aureus as you see it. And so I'm going to get out of this area. This area right here is too thick. That's why you want to avoid making thick smears. This area out here is still thick, but we're starting to resolve individual cells and clumps of cells. Let's recall the Latin name Staphylococcus. Staphylo means grape-like clusters. That refers to the arrangement of these cells so that they do look like giant clumps of grapes. And coccus means spherically shaped cells. Of course, when you look at a slide like this, you're going to see cells that have fallen away from the clumps. You're going to see cells that may look like short chains but are in the process of dividing. In fact, to see the cell arrangement of Staph aureus, it's a little easier to see that on the simple negative stain. So we'll take a look at that in a moment. But for now, I'm going to show you, if you move through your slide, you always want to canvas your whole slide. You don't want to look at this and say, oh, I did a bad job. Nope, you can always go to the edge, tweak your fine focus knob, and you can find an area that is less dense. Even on the densest of slides, if you go to the edges, you will generally be able to see a thinner smear. Okay, so what do we learn from this? First of all, did crystal violet help us see Staph aureus under the microscope? I would say yes. We have this tremendously white background and now we have dark purple cells. So we can see that the shape is a coccus. We can see some arrangements here that are grape-like in their clustering. Let's compare this on the same slide, and for this I just need to slide over to this region. On the other half of the slide, fine focus, and now all of a sudden what do I have? I don't have cocci, I actually have short rods. These short rods, some of which look like little eggs, they're almost cocci, but they're known as coccobacilli. These are actually E. coli. E. coli, it's debatable whether you want to call it a short rod or a coccobacillus. But the thing about E. coli is you can see very obviously in no way, shape, or form is it a perfect sphere like Staphylococcus is. So it may look flat tire-ish or it may look nice and rod-like. It depends on the stage of growth and whether we are looking at two cells in the act of binary fission. As you can see right here on the screen, they are dividing in two, and the cell length can vary as the cell grows right before it gets ready to divide in half. But we've got a really nice view of E. coli. I would like you to stop the video and draw E. coli in your lab manual. I'm going to remove this slide, and we're going to take a look at Bacillus subtilis and Rhodospirillum rubrum. So again, this is with crystal violet. I simply slid the old slide out. I have oil droplets already on the other slide, and I moved that in. Now what I need to do is tweak the fine focus knob and look at what we have. And we have some movement there, and you say, oh wait, I thought she said she killed those cells when she heat fixed them. In fact, I did, but what you are seeing is the Brownian motion because there's still a little water left on the slide, and that water mixing with the oil is giving us the sensation of motility. Okay, so when we look at Rhodospirillum rubrum, that's pretty dense. That is a dense smear. You can see that the cells are longer than E. coli and they appear curved. If you grow Rhodospirillum rubrum out long enough, I'm just going to scoot it. Let's see if I can scoot it to the edge and find an area not quite as dense as the others. If you grow it for a long enough period of time, it actually forms the letter S. In fact, it can have multiple waves in it, not just two. So we can look around here, and I found a cell that looks like a spirillum. So let's take a look right here. Do you see the S shape? That's Rhodospirillum. Here's another S shape. So here's another S shape. That is a spirillum. Okay, so we see the shape of Rhodospirillum. Its name told us ahead of time what we were going to see. We see that just like E. coli and Staph aureus, it is purple because we used one dye, and that dye was called crystal violet. Now, on this slide here, I have Bacillus subtilis. And I want you to see these dark purple, long, thick rods that look like sausages. That is what we call a vegetative cell of Bacillus subtilis. In addition, if we look around, we start to see some egg-shaped structures. And at times, we see that some of these long rods did not take up the crystal violet as well. That is because these are starting to die. And as Bacillus subtilis undergoes different environmental stresses, it begins to sporulate. In lecture, we will talk about spores, but I want you to look at this cell right here. Do you see how it's purple around the outer edges, but inside it's lavender? Here's an example here, here, here. These are egg-shaped spores. Over here at the bottom left of the screen, you see egg-shaped spores. Here's a spore. Not to be confused with the vegetative rod-shaped, thick sausage or hot dog-like cells. So there are times when the cell begins to sporulate and the spore is actually seen inside the still vegetative cell, and there's other times when the vegetative cell has died, degraded, fallen away, and thus it is not taking up the dye, and what's left are the spores. So this is a mixed culture. This culture was getting old. It in its major growth stage, all you see are dark, long, thick rods. But as it uses up all of its nutrients and begins to suffer from the diminished nutrient availability and the increased waste, then it sporulates and it forms these egg-shaped structures that do not stain all the way through. So here's a cluster of spores. Okay, so that's Bacillus subtilis. Here's a great view, and this is what I'd like you to draw in your lab manual. You can see many vegetative, thick, hot dog-shaped rods Draw those and make a note that they are vegetative. That is a term that means actively metabolizing. And then we also have the spores I want you to draw. And there may actually be some examples where the spore is actually forming inside the rod while it begins to decay. And you may draw that as well. Okay, so those are our 4 bacteria. Bacillus meant rod-shaped. Let's just very quickly take a look at our simple basic stains where we use the dye known as safranin instead. I just want to show you the difference in color. Safranin is pink. So what you are seeing here on this slide is Staphylococcus aureus and its grape-like clusters of ball-shaped cells, and it is pink, not purple. If I scoot over here to E. coli, we should see that E. coli also stained pink. Got to find our E. coli cells, and let's see. Here's a very thick cluster. That is not a good smear. So what do? You go out to the edges and there you find the pink rods. Compare that to those dark purple that we saw when we dyed them with crystal violet. Okay, so we've seen Staph aureus and E. coli stained with both crystal violet and safranin. Let's take a very quick look at Rhodospirillum rubrum and Bacillus subtilis stained with safranin. Let's see if I can move into my smear. There we go. And you see those lovely S-shaped, or at least comma-shaped if they're not long enough, Rhodospirillum rubrum. Just lovely spirilla. Look at that. Perfect S, perfect letter S. Okay, so that's Rhodospirillum rubrum with safranin, nice and pink. And we have Bacillus subtilis. Bacillus subtilis, although the rods are slightly pink here, we have a lot of hollow egg-shaped spores that you see on the screen that they don't appear to have taken up safranin very well. Let me get it, there we go. Get the color altered here just a little for you. So spores, which are egg-shaped, do not in fact take up dye well. So you end up getting a lot of what look like hollow eggs here. But you can still see some of the vegetative pink rods of Bacillus subtilis, a long, thick bacillus. Okay, that brings us finally to our simple negative stains. Let's recall with the simple negative stain, we did not in fact heat fix these slides. We simply put a drop of nigrosin on these slides and we we smeared it across the edges in order to spread the cells out over the slide. Now, what I'm doing here is I am avoiding the pitch black background of that slide. We use nigrosin to block out the white background. So we are simulating dark field microscopy without needing those special plates and filters under the stage. And so what you do when you use a simple negative stain is you go to a lavender gray area of the slide, not the real thick nigrosin because you won't be able to get the light of the microscope through it. And here, what it allows us is a view of Rhodospirillum rubrum where we get an idea of the true shape of the cells because the nigrosin is negatively charged and it has repelled rather than stuck to our Rhodospirillum cells. So since they are also negatively charged, the nigrosin stays in the background, occluding the slide. The cells repel the nigrosin and appear white. So look at these beautiful spiral cells. Okay, so let's just move around here a little bit and have a look at areas of the slide where it might not be as nice of a smear. So you look here and we're starting to get pretty dark. So dark that even the cells are not illuminated. So you always, when you look at a slide that has a nigrosin smear on it, you don't go to the darkest spot. You go to the edge where you see the gray or lavender area, and that allows you just enough of a contrast between the cell and the background to see the beautiful shapes of bacteria. This is just a really nice slide. Go ahead and stop the video and draw what you see here on this slide. So one of the learning objectives I had for today is that you learn the proper Latin for cell morphologies and cell arrangements. We know that Rhodospirillum, the cell morphology, that means the individual shape of each cell, is a spirillum, an S-shaped or spiral-shaped bacterial cell. But what is the cellular arrangement here of Rhodospirillum? By cell arrangement, I mean when the cells undergo binary fission, do they split apart and survive as individual cells like Rhodospirillum? If so, we call that a single arrangement. Rhodospirillum rubrum, Bacillus subtilis, Escherichia coli, those were all single arrangements. Some species divide in two and they stick together as a pair of cells, and we call that a diplo. Some keep dividing along that plane and they create a long chain called strepto. I'm going to trade this slide out for the simple negative smear I made of Staph aureus. And recall that Staphylococcus aureus now is a ball-shaped cell in a grape-like cluster. That's what staphylo, S-T-A-P-H-Y-L-O means, staphylo. And here, on a simple negative smear, you get to see those grape-like clusters and resolve the individual cells within the cluster. And that's why this is such a powerful, simple stain for staph here. It allows us to not only note the cell morphology being coccus, but the cell arrangement being staphylo. And that is a lovely view. So let's go ahead and stop the video, and what I'd like you to do is draw some of these staphylococcal clusters in your lab manual showing the individual cells. With Staphylococcus aureus, the cells divide in two along all three planes, the X, Y, and Z, and they stick together, they stay together. They're not, they're still unicellular, but they simply don't pull apart after binary fission. So this just gives you that idea of the staphylo arrangement. We have for arrangements single, diplo. If it divides on two planes and stays together as four cells, we call it tetrad. We have strepto, which is in chains, and we have staphylo, which is in grape clusters. There are also other terms such as sarcini. Sarcini means in a cube-shaped cluster of 8, 16, 32 cells, creating almost a box-like structure, kind of squared off in its arrangement. Okay, so that does it. We have completed our simple staining. Let's just recap very quickly. We did simple staining using basic dyes, and we called that a simple basic stain. We used either crystal violet or safranin, both of which are positively charged. Thus, they stuck to the cell wall and cell membranes. The surface of the cells adhered and provided contrast between the cell and the bright field background. When we made these simple basic smears. Recall that you don't want to overwhelm a loop full of water with bacterial cells. You want to make a loose arrangement so that you can see individual cells, but you want to make it thick enough so that you leave a smear that you can basically see through, but you can see is slightly cloudy. When you're done making that smear, you have to allow it to air dry. Once it's air dried, you heat fix it by passing it quickly through a flame 3 or 4 times. This allows the cells to stick to the slide so they do not wash away when you add the dye. You add the basic dye, it sits for a minute or 2 minutes, you rinse it off with water, you towel blot the top, you wipe the bottom, put oil on it, and voila, you're ready to go. With a simple negative stain, we took a drop of nigrosin, we put our bacterial inoculum in that drop, and we used a second slide to spread it across the surface. We let that air dry. We do not heat fix simple negative stains, and the nigrosin is negatively charged. It repels the negatively charged cell surface, and it allows us to highlight those individual cells and to better get an idea of their cellular arrangements. Okay, that's all for now. I'll see you next time in session 6.